Looking at this slide of fish mitosis, we can use our image from the SLM that has the nucleus at the bottom to compare to the nuclei on our slide. And we can see on this slide of the fish, all of those nuclei appear to be the same. They're still round. They maybe have some granules or some lobes within the nucleus that we can see, and most of them appear to all be prophase. Let's look at a onion root. So this is a plant which has cell walls. So the cells are much more organized, in a square-like or rectangle-like pattern. But looking at the nuclei, we can see most of these nuclei appear to be pretty circular. We can see a little bit of granules or lobes within the nucleus. And most of the nuclei and cells on this slide appear to be prophase. Let's look at metaphase, and using the picture from the SLM, we can see how the nucleus should appear at the bottom of that image. Looking at a fish cell again, can you find the two metaphase cells on this picture? It appears to be these two. We can see their central nuclear area. We can still see the little bit of a dark coloration, but it's not a circle. It's kind of a straight line across the middle, and these ones are oriented slightly different. Just to give you an idea of, this is a three-dimensional object. Let's look at the plant cell. So again, we can see the nuclei. Which nuclei are aligned in a line at basically the equator or midpoint of the cell. And again, there are two examples of metaphase in this image. Are you able to find them? Those two right next to each other? Not perfect, but generally all of those chromosomes are in the middle. There is some overlap, some sticking out just above and below the plane, but most of them are directly on that midline, and we can see them as metaphase in this fish cell example. Moving on to anaphase. Again, we have our SLM image and a fish cell image. Looking at the bottom of the SLM image, we can see that our chromosomes have begun to pull apart from that metaphasic plane and are beginning to move towards the polar ends of each cell. There are two examples of anaphase on this slide. Are you able to find them? And you can see both examples. We can clearly see the chromosomes pulling away from them. There is a definite gap in space, and they are moving towards the opposite polar ends. Looking at the onion cell, or the plant cells, we can also see two examples of anaphase on this slide. Are you able to find them? We're looking for chromosomes that have been separated, and these two. They've been pulled apart. They're obviously away from the metaphasic plane towards the polar ends, and they have not moved on to telophase yet because we do not see that cleavage furrow. So these are still within anaphase. Telophase is the final stage of mitosis and can be either the most difficult or the easiest to identify based on your ability to see a cleavage furrow. So if you look at the example of the nuclei in the SLM image, we can see the nuclei are pulled the whole way to the ends and actually begin even maybe reforming into the new nucleus. And looking at the diagrammed example, we can see how that there are two lobes, one half on one side, one half on the other side, and they combine where that plane of constriction, and that is the cleavage furrow. So looking at this fish slide, are you able to find the cell that has the cleavage furrow, where the chromosomes are just barely connected through the opening that is left? This cell that will soon become two cells is in telophase. we can see the nuclei still appear just to be connected through the middle of that cell. But whenever I remove the circle again and you follow the cell membrane, there's an obvious indentation kind of between those two cells where those nuclei are connected, and that is the cleavage furrow. Let's look at the plant cell. So the same thing, we're looking for a cleavage furrow or a line to form in between. Now this is much more difficult to see on the plant cell sometimes because the softness of the animal cell allows that constriction or that plane of constriction to occur that we can see. The rigidity of the cell wall in the plant cells mean that we get a pencil line kind of starting in the middle that will slowly make its way to the edges of the cell, eventually splitting them into two separate cells. So can you find the cell here that kind of is almost separated but not quite there in telophase? It's this cell. We can see some spindle fibers maybe on the edges of the nuclei, and then the kind of line in the middle just isn't thick enough and just barely doesn't seem to get the whole way to the cell walls, making that into two separate cells. So that's our example of the cleavage furrow and the plane of constriction in the plant cell. Let's try an example without the image to help us. And there are multiple phases of mitosis very close together in this slide. Can you find the metaphase? Can you find the anaphase? And can you find the telophase cytokinesis? And it actually appears that one and this cell are both in the telophase. We can see the cleavage furrow and the cells just barely connected sort of in the middle. Let's do an example with the onion cells without the SLM picture there to help us. There are several different phases of mitosis on this slide. Which ones can you identify? Can you identify metaphase? This one, but there's also maybe the other one directly to the diagonal upper right of it as well. What about anaphase? And what about telophase? Are there any in here that you see that are very close or appear to be in telophase? What about this one? I think I can just barely start to make out a thin line right across the middle of that. Two cells in between the nuclei. And that's what we're looking for. That's that plane of constriction or that cleavage furrow.